So as a group, we decided to focus our research topic on Duchenne muscular dystrophy and looking at comparative genes of different models within the disease. And I'm here with Liz and Dan, and we will explain that to you.
Okay, so Duchenne muscular dystrophy is one of the most common forms of muscular dystrophy and is caused by a recessive mutation in the dystrophin gene located on the X21 chromosome. And then dystrophin is important in the study and growth of muscles, and without it, muscles progressively get weaker and weaker, causing the males to typically die in their late 20s to early 30s. We hypothesized that by finding the mutation in the gene, we could implement gene therapy to substitute the defective gene with a functional dystrophin protein, putting a complete stop to the progression of muscle weakness that is not due to old age. And although this would completely stop any progression, it does not take away from the fact of any cause or harm that it had already made in the individual. But there are other things like steroids or physical therapy that will help with that.
So the research topics and questions that we want to focus on within the topic of DMD was finding statistically significant differently expressed genes or DEGs between healthy and DMD diagnosed genomes, as well as what genes can we find that can be used for research as targets for treatments and therapy, as well as we looked at genes between mice and human to see how comparative they were and to see if there were any significant genes between the two to determine if mice can be used as an animal models to treat humans within the pharmaceutical manufacturing process. So the way we found our information is we use BLAST as a tool to compare the genomes between humans and the rat genome, and which calculated data as well. And that uses a bunch of different algorithms to do that. And then we moved on to using Cyverse, which is a tool considered for RNA sequencing. It already had the human genome, and we were able to upload DMD genome and another specific genome within it that found upregulation, downregulation within different genes among each genome. So it was able to measure what was being upregulated more and what was being downregulated more compared to the healthy genome. And then once we had that information, we were able to upload it into our studio to create a volcano plot that was able to show us on a logarithmic scale what genes were significantly expressed and which ones weren't. And then we then use that for more research and more results.
So as previously mentioned, we used BLAST to compare different genomes. Especially, we were interested in kind of the CRISPR aspect of genetic modification as a treatment. This shows the BLAST results comparing dystrophin in humans to mice. The e-value works the same way as a p-value. The lower the value, the better. And they all have 0.0 between dystrophin in mice and humans and the eutrophin in humans and mice, because that says that there is no expected better fits. That's what the 0.0 means. This is just farther explaining the importance of genetic parallelism. And this shows how the CRISPR-Cas9 works in mice and how it would work in humans if there is parallelism, which there is in like a carryover experiment. So this is the volcano plot we generated in R. The green represents no change in upregulation when we compared samples from regular or patients that don't suffer from DMD to DMD sufferers. The red represents upregulated genes or downregulated, and the blue shows upregulated genes. So we found a total of 6. And of those 6, just gotta move the screen so I can see. Of those 6, two were pseudogenes. So a pseudogene essentially is an imperfect, non-functioning copy of a functional gene. So it doesn't work. It's like it's just all wrong. You can't. It doesn't code for proteins. And the other one is an LINC gene. So that is a gene which its length is exceeding 200 nucleotides, and it's not translated into proteins. Its main use is just for transcription regulation. Of the DFGs we did find, the most important ones. The most important ones were seen to alter T cell production, and they have high correspondence in protein receptors and their functionality. It's important to compare DFGs of like when they're upregulated and downregulated because it shows what's different between a normal genome and someone that has a disability or like a dysfunctional genetic condition. So it pinpoints exactly what the differences are. So development of a genetic treatment will show you where to pinpoint and where to start from.
All right, and that is majority of our project, and we hope to explain more during the virtual Kirk event. Thank you.